Assalamualaikum friends, my name is OMAMA SHAH and today I will be discussing fatty acid oxidation. We will see when a fatty acid oxidizes how much ATP it generates, and there will be questions at the end for your better understanding. My channel name is OMAMA SHAH MEDICOS. If you like my videos, must subscribe to my channel for further new uploaded videos. Before starting the fatty acid oxidation, I would like to build up your concept regarding fatty acid. So fatty acid is basically a component of lipid, and there are different types of lipids. Now we are going to discuss. The first one is the acyl glycerol. The common acyl glycerol is a triglyceride which contain one glycerol molecule and 3 fatty acids. The second type is the phospholipid, which contain one glycerol molecule, 2 fatty acids and 1 phosphoric acid. The third type is the waxes. The waxes contain long chain fatty acid bound to long chain alcohol. The fourth is the steroids. Steroids are the only lipids that do not contain fatty acids. Why we do fatty acid oxidation? We do it because the body needs energy in the form of ATP, but there is not sufficient glucose present. So in the state of starvation, the body oxidizes fatty acid in order to generate ATP. Fatty acid oxidation. The oxidation of fatty acids will take place in a state of starvation. The fatty acids in the body are mostly oxidized by beta oxidation. Now let's see what beta oxidation is. When an oxidation takes place on the beta carbon atom, that oxidation is called as beta oxidation. The beta oxidation results in the removal of a 2 carbon fragment, and that 2 carbon fragment is called as acetyl coenzyme A. The acetyl coenzyme A produced as a result of beta oxidation will be completely oxidized to carbon dioxide in the citric acid cycle, and this will conserve the energy as the body is in the state of starvation. So the conservation of energy is in high demand. Maximum oxidation takes place in different organs that are written. Minimum or zero oxidation takes place in the following organs that are written: heart muscles, skeletal muscle and liver. In these organs, the oxidation will be maximum, while in brain, erythrocytes and adrenal medulla, there will be minimum or zero fatty acid oxidation. In liver, when the acetyl-CoA is produced by beta oxidation, it may be converted to ketone bodies, which are then transported to the brain or other tissues where there is deficiency of glucose, as because the body is in the state of starvation. How many stages are there of fatty acid oxidation? There are 3 stages of fatty acid oxidation. They are activation of fatty acids in cytosol. So first we will activate the fatty acids in the cytosol. Then what we will do next? We will then transport it into mitochondria. So then what will we do? Then we will oxidize the fatty acid, and the cut will be on beta carbon. Therefore, that oxidation will be called beta oxidation. That cut will remove 2 carbon fragments. That 2 carbon fragments is called acetyl coenzyme A. So now let's discuss each of them one by one in detail. Now we are going to discuss the first stage of fatty acid oxidation, such that activation of fatty acid. The fatty acid is said to be activated when it is converted into fatty acyl coenzyme A. The fatty acid is converted into fatty acyl coenzyme A by 2 steps. In the first step, the fatty acid combines with ATP and form acyl adenylate. In the second step, acyl adenylate combines with the thiol group of coenzyme A and form acyl coenzyme A. These 2 steps are performed by an enzyme called fatty acyl coenzyme A synthetase. As synthetase is for synthesis, synthesis means formation. So formation of fatty acyl coenzyme A takes place by fatty acyl coenzyme A synthetase. Here you can see from the figure, this is a fatty acid and this is ATP. The ATP combines with fatty acid by an enzyme fatty acyl coenzyme A synthetase. It is converted into fatty acyl adenylate. The thiol group of coenzyme A then takes the fatty acyl adenylate and convert the structure into fatty acyl coenzyme A. This first step requires ATP, and here AMP is removed in the second step. The reaction completes when a pyrophosphate is hydrolyzed into 2 molecules of inorganic phosphate by an enzyme inorganic pyrophosphatase. As you can see over here is the minus sign. This indicates that the reaction is highly exergonic. So friends, now we will transport the activated acyl coenzyme A into mitochondria. This is the second stage in fatty acid oxidation. It has 2 steps. In the first step, the acyl coenzyme A is converted into acyl carnitine. Carnitine is added to acyl coenzyme A while coenzyme A thiol group is removed. This step is performed by an enzyme called carnitine acyl transferase 1, and this step occurs in the cytoplasm. In the cytoplasm, the acyl coenzyme A is converted into acyl carnitine by addition of carnitine and removal of coenzyme A thiol group. The step is performed by an enzyme called carnitine acyl transferase 1. Now once this is done, the acyl carnitine is transported into the mitochondria, and there the second step occurs. In the second step, the acyl carnitine is converted into acyl coenzyme A by the addition of coenzyme A thiol group and removal of carnitine. This second step is performed by an enzyme called carnitine acyl transferase 2. As I discussed earlier, that this second step occurs in the mitochondria. Now then in the third step, we will perform beta oxidation on this activated acyl coenzyme A. Here you can see from the figure, this is acyl coenzyme A. With acyl coenzyme A, a carnitine is added and formed acyl carnitine, while the coenzyme A with thiol group is removed. This reaction is performed by an enzyme called carnitine acyl transferase 1, which is present on the outer mitochondrial membrane. Once acyl carnitine is formed, it is transported into the inner mitochondrial matrix through inner mitochondrial membrane. Acyl carnitine in the inner mitochondrial matrix is converted into acyl coenzyme A, which is called as activated fatty acid, by the addition of coenzyme A with thiol group and removal of carnitine. This carnitine returns back to the cytoplasm for its reuse. So this was all the mechanism by which the activated acyl coenzyme A is transported into the mitochondrial matrix by carnitine shuttle, such that carnitine carrier proteins. Now coming towards the last step of fatty acid oxidation, such that beta oxidation proper. This is the last and third stage of the fatty acid oxidation. It involves a sequence of 4 reactions. First one is the oxidation, such that 2 hydrogen atoms will be removed. Second is the hydration. There will be addition of water. Third is again oxidation, and 2 hydrogen atoms will be removed. And the last step will be the cleavage, in which acetyl coenzyme A will be removed and formed, and then will enters into the Krebs cycle for further oxidation. Now we are going to discuss the first step in beta oxidation proper, and that is oxidation. In oxidation, acyl coenzyme A is converted into delta 2 trans-enoyl coenzyme A. FAD is reduced into FADH2 by an enzyme, acyl coenzyme A dehydrogenase. In this step, double bond is formed between alpha and beta carbons. Now we are going to discuss the second step in beta oxidation proper, and that is hydration. In hydration, the delta 2 trans-enoyl coenzyme A that was product in the first step now is a reactant in the second step. It is hydrated and converted into beta hydroxyacyl coenzyme A by an enzyme, enoyl coenzyme A hydratase. It is to remember over here that enoyl coenzyme A hydratase brings about the hydration of the double bond to form beta hydroxyacyl coenzyme A. Now we are going to discuss the third step in beta oxidation proper, and that is again the oxidation. In oxidation, the beta hydroxyacyl coenzyme A is converted into beta ketoacyl coenzyme A by an enzyme, beta hydroxyacyl coenzyme A dehydrogenase. All other word is the same as beta hydroxyacyl coenzyme A, just you have to enter dehydrogenase, such that here the removal of 2 hydrogen atoms will take place and NAD will be reduced to NADH and hydrogen positive ion. It is to be noted that beta-hydroxyacyl coenzyme A dehydrogenase catalyzes the second oxidation and generates NADH. The fourth step in beta oxidation proper is cleavage. Here, the beta-keto-acyl coenzyme A will be converted into acyl coenzyme A with 2 carbon less by an enzyme thiolase. Coenzyme A with a thiol group is added in this fourth step of cleavage, and we receive acetyl coenzyme A as a product. This acetyl coenzyme A will either enter the Krebs cycle and do further oxidation, or it will convert into ketone bodies. Beta oxidation. Here I can summarize it in short. Here the fatty acid used is palmitic acid. It is first activated by addition of coenzyme A. It is then oxidized into trans delta 2 enoyl coenzyme A. This process is called oxidation because here 2 hydrogen atoms are removed and FAD is reduced to FADH2. The enzyme acyl coenzyme A dehydrogenase catalyzes this oxidation reaction. In the next step, trans delta 2 enoyl coenzyme A is converted into beta hydroxyacyl coenzyme A by an enzyme enoyl coenzyme A hydratase. Here the enoyl coenzyme A hydratase brings about the addition of water, and delta 2 over here means that there is double bond between alpha and beta carbon atoms. This process is called as hydration because here the water is being added. The beta hydroxyacyl coenzyme A is then again oxidized to beta ketoacyl coenzyme A by an enzyme beta hydroxyacyl coenzyme A dehydrogenase. All the name is same of beta hydroxyacyl coenzyme A, only with enzyme you have to put dehydrogenase. Whenever the enzyme came dehydrogenase, there will be a removal of hydrogen atoms, and either NAD or FAD will be reduced to FAD or NAD respectively. Over here you can see in the third step, NAD is reduced to NADH and hydrogen positive ion. The beta ketoacyl coenzyme A in the fourth step is then cleaved to acyl coenzyme A, which has 2 carbon less than the original palmitic acid, which was having 16 carbons, and acetyl coenzyme A, which is a 2 carbon compound. Here the addition of coenzyme A with the thiol group is aided by an enzyme thiolase. The cycle will proceed until there are 7 rounds completed. When the 7 rounds are completed, we get 8 acetyl coenzyme A. These acetyl coenzyme A will either enter the Krebs cycle for further oxidation, or they will be form the ketone bodies. So this was all about its brief summary. Here you can see the overall reaction. These are the reactants, while these are the products. Acyl coenzyme A, carbon number may differ. It may vary. It may be either 16, 32, depend upon the fatty acid used. FAD, NAD, H2O and coenzyme A with the thiol group is in the reactant side. The product we will get acyl coenzyme A, which will have 2 carbon less than the original one, because these 2 carbons are now in the form of acetyl coenzyme A, which we get the product, and FADH2, which is the reduced form of FAD, and NADH, which is the reduced form of NAD with a hydrogen ion. Key points of the topic. Acyl transferase 1 is inhibited by malonyl coenzyme A, which is the first intermediate in fatty acid synthesis. Formation of each acetyl coenzyme A requires removal of 4 hydrogen atoms, means 4 electrons and 4 protons, from the fatty acyl by dehydrogenases enzymes. Acetyl coenzyme A formed in the liver during oxidation of fatty acids can either enter the citric acid cycle or undergo conversion to the ketone bodies. Now when a fatty acid is being oxidized, let's see how much ATP it generates. We will take an example of fatty acid, palmitic acid, which has 16 carbon. Now it will complete 7 rounds and 8 acetyl coenzyme A will it form, 7 NADH and 7 FADH2 will be produced. The 8 acetyl coenzyme A will enter the Krebs cycle. 1 acetyl coenzyme A will give us 3 NADH, 1 FADH2 and 1 ATP. So the 8 acetyl coenzyme A will give us 24 NADH, 8 FADH2 and 8 ATP. Now net generation of ATP. Here 24 plus 7. The 7 is from here 7 NADH and the 24 we get over here. That 8 acetyl coenzyme A gives 24 NADH, while in beta oxidation we get 7 NADH. So we add 24 with 7 and we get 31 NADH. This 31 will be multiplied by 3 because 1 NADH gives 3 ATP. So 31 will be multiplied by 3. So 31 NADH will give us 93 ATP. 8 plus 7. This 8 is over here. In Krebs cycle we get 8 FADH2 while the 7 is over here. In the beta oxidation proper we get 7 FADH2. When we add this we get 15 FADH2. As we know in electron transport chain 1 FADH2 gives 2 ATP, so 15 multiplied by 2 gives 30 ATP, means 15 FADH2 will give us 30 ATP. Eight ATP are produced in the Krebs cycle, so 8 we get 8. Now we will count these. 93 plus 30 plus 8. We get 131. So when a palmitic acid is oxidized, we get 131 ATP. Questions of the topic. If you know the answers of these questions, must answer it in the comment box. Allah Hafiz for now.